honor to introduce Cynthia Michaud, who is defending her master's thesis today. And so I'm just going to tell you a little bit about Cindy so that you know where she came from before she came to Moss Landing. She grew up in Methuen, Massachusetts. And I think it's actually really important that you all know that Cindy is from New England. One time I accidentally made a disparaging remark about the New England Patriots, and I'm not sure I've regained her respect since then. Cindy graduated from St. Monica's Elementary School, and then a few years later, she enrolled at the University of Rhode Island, and I think that's where we can start to see her interest in a lot of different disciplines in science. She majored in both marine biology and geological oceanography, and she discovered her love of phytoplankton when she started working with Susanna Mendendoyer. She went out to sea and did dilution experiments and then worked in Susanna's lab. And I like that it's kind of come full circle because now she's using some really important equations that Susanna derived for her master's thesis. And she also participated in an REU at the Bigelow lab with Mike Lomas. And there she was studying nutrient uptake by phytoplankton. And she was able to present that research at an ASLO meeting in Hawaii. And then in 2017, so just a little over 2 years ago, Cindy came to Moss Landing for her master's. And here, I think you'll see, especially from her thesis research, that she's continued to pursue these interdisciplinary interests. She was the TA for both biological oceanography and physical oceanography, which I think is pretty remarkable that you could TA both of those classes. She worked at the iTech help desk for a year. She was also the student body treasurer, and she received a WAVE award. Besides doing her master's research, which you will hear about today, she also worked with Tom working on the California HAB map database, which was basically, she took Jason Smith's data, monitoring data from the wharf, and made it more publicly accessible. Then she also did work with Tom studying the seasonal cycle of deep ocean currents, and so what she did with that project was analyzing current meter data. So you can see that Cindy is kind of like a triple threat here. She's got the biology, taxonomy expertise, she's got the physics, and she has this background in geology. And actually that work that she did with Tom led to a co-authored paper that she's on. And then the work that you're going to hear about today was supported by the California Sea Grant. And again, I think you'll see her interest in both. She's incorporated mineral analysis into her work. She's become a phytoplankton taxonomy expert. And then she also, you'll see that she's still thinking about the physics too, and how that relates to her results. So now I'll hand it over to Cindy and let you tell us all about your research.
All right, well, thank you all for coming. I'll be talking about my thesis, which is the effects of phytoplankton composition and biominerals on the episodic pulses of particulate organic carbon to abyssal depths. So I want to give you a background on the biological pump. So here you see the phytoplankton growing at the surface of the ocean. They're taking carbon from the seawater, and they can bring their carbon down to the deep ocean through two forms. One form is aggregate formation, where you have the phytoplankton growing together. They form an aggregate, and then they bring their carbon down to the deep sea. Another way is through grazing by zooplankton. The zooplankton will form them into fecal pellets. They get packaged up in the fecal pellets, and they bring their carbon down to the deep sea that way. So why is the biological pump important? So this is a figure from the IPCC 2014 report of the total carbon cycle the carbon budget of the global carbon budget. So I want to point out these two arrows. So this arrow is showing the total amount of carbon that humans will produce per year, and this arrow is showing the amount of carbon that is sinking from the surface to the deep ocean. So humans will produce around 7 petagrams of carbon per year. Well, here it says that there's 11 petagrams of carbon that will sink from the surface to the deep ocean, when actually it's around 5 to 12 petagrams. And I'd like to point out that this 5 to 12 is an error around 7, which is the same amount that humans can produce per year. So we don't know how much is actually sinking, how it's sinking, and the importance of it. So I'd like to go over this paradigm of how is this carbon sinking. We know we have a lot of carbon at the surface, but as these aggregates sink, they get degraded by bacteria, they start to attenuate, the carbon starts to attenuate, and when they reach down, only a small amount of the carbon makes it down. Same thing with the zooplankton. When they get grazed upon, their fecal pellets start to degrade, and the carbon that's packaged in the fecal pellets will attenuate and won't make it all the way down to the bottom. So this is important in understanding how much carbon is making it down for carbon sequestration to the bottom of the ocean, but also for the benthic organisms that live down there. These benthic organisms rely on the carbon source as their food, and I have this video from Mbari that shows the benthic organisms that live down at the deep ocean. And if you look around, you can see all the little organisms on the bottom, and then at the end, you'll see a big pulse of carbon that's sinking, and that is the carbon going down. So I'll let you watch that. All right, so how do we measure the carbon down to the deep ocean? Here is an example of the sediment trap. So this sediment trap is a large funnel-shaped instrument that will collect the water that is sinking down to the deep ocean. And the way that these things work is that these are sample bottles that are attached to a carousel here. And every 10 or 17 days, these will close and move over to the next one. So that's how we get the samples that we'll be looking at for the carbon sinking. So an important station of where we collect the carbon is at Station M. So Station M is a long-term oceanographic station located off the coast of California. And this station is part of Ken Smith's Mbari's research that he's been doing. And what's really important is that this is a 30-year time series that hasn't been really looked at for biological aspects of it, but they've been looking at the POC that's sinking and trying to correlate it with satellite remote sensing and seeing how the chlorophyll and the phytoplankton will sink down. I also want to point out that this trap right here is the trap that I'll be looking at for my samples, and I just want to point out the benthic rover. I will show you a video from the benthic rover in a few minutes, but the benthic rover moves along the sea floor and takes pictures of what is down there. So looking at the time series from Station M, this is the 30-year time series of POC, so particulate organic carbon. And what I want to point out are the red spikes that you can see. Those red spikes are short-term episodic pulses of organic carbon. So what I wanted to see is what was causing the difference in the episodic pulses of particles in these high flux events compared to the normal flux in this area. And I also want to point out that these large episodic pulses can attribute between 25 to 63% of the annual carbon export in this area. So that benthic rover I talked about, this is a time-lapse video showing the images from the benthic rover is showing fluorescence. So all the lit up things that you see on the bottom of the seafloor is fluorescence. So this is important because phytoplankton will have the fluorescence. So being able to see that their phytoplankton sinking to the deep ocean is important for this study. So now that we know that phytoplankton are important in making it down to the deep ocean, here is a satellite image from NASA showing a large phytoplankton bloom off the coast of California. So this is a June-July 2017 composite, and I just want to point out that this bloom corresponds to one of those high flux events that I had shown you on the total POC flux. So we see that there's a large phytoplankton bloom. So why is there so much phytoplankton growing here? So we know the California current is a big place for upwelling. So what is upwelling? Upwelling is when surface winds are pushing water from the coast offshore. So when the surface water gets pushed offshore, cold, nutrient-deep water will rise up, bringing it to the surface, and this will allow phytoplankton to bloom at the surface. So if we look back at the satellite image, we can see that the upwelling is occurring right along the coast, bringing large nutrient-rich water and allowing these phytoplankton to bloom. Now looking closer at this, we see that this is where the upwelling along the coast is, but we have this front that's occurring here. So there's been studies that show evidence of increased carbon export at the fronts and through subduction. So subduction is when there will be water that is sinking rapidly and pulling the phytoplankton and other surface organisms down to the deep ocean. So it's important to now look at the types of phytoplankton that we can see off the coast of California. So this is a graph from Venrick, which is from the CalCoffee data that shows the ship transects that show the different types of phytoplankton off the coast of California. And what I want you to see from this graph is that these 3 bars are coastal waters, while this one's an offshore water. And the main takeaway here is that the coastal waters have a majority of diatoms. So over 50% are diatoms in the coastal water. Well, on the offshore waters, you have a majority of Coccolithophores with less diatoms there. So looking back to the satellite image again, this water right here would be diatom rich, while this water over here would be Coccolithophore rich. So if you have this diatom rich water moving offshore and this increased export here, we might have more diatoms that are relating to the export. So bringing it back to this station M time series, we see that there is an increase of total POC as you get closer to present day, and we do see these increase in episodic pulses. So now I want to talk about some potential mechanisms that could be causing this increase in total POC. So my first mechanism I want to go over is the aggregation of large diatoms along with the physics of the California current. So here I have these large, slow-growing diatoms. I know I mentioned before that phytoplankton like to grow in nutrient-rich waters with upwelling, but these are certain types of diatoms that grow at the base of the euphotic zone. They like low light intensities. So these are large, slow-growing diatoms. When they get disturbed, they get pushed up to the surface. They don't like the higher light intensity, so they get stressed out. When they're stressed out, they form carbohydrates, become sticky, and form these large aggregates. These aggregates will then sink down to the deep ocean. So my next mechanism I want to go over is ballasting and biominerals. So here I have phytoplankton again, but I tried to make these cell walls thicker to show that they're enriched with biominerals. And so these biominerals, when they get eaten by zooplankton, they'll make their fecal pellets and the particles that they're sinking in more denser, and they will sink down. So what are the biominerals and ballasting? So the 2 biominerals I'm looking at are biogenic silica, which comes from diatoms, and particulate organic carbon, which comes from Coccolithophores. And what I want to say is the Ballast Hypothesis is that these cells that contain the biominerals are denser compared to the surrounding waters, and they will sink more quickly when they're in particles because they're ballasting these biominerals. And so when I look at this figure here from Clausen Archer, this is a figure that supports the ballast hypothesis, that as you increase POC flux, you have an increase in the biominerals. So here we have opal, which is the biogenic silica. It's from diatoms. And on this graph, we have calcium carbonate, which is particulate inorganic carbon, which comes from Coccolithophores. So they both have a positive linear correlation with POC. And I also want to mention that this study was done globally, and it has different seasonal averages, and it's sampled all over the world. So another important point to think about with ballasting is the limitation of iron. So I want to orientate you with this graph. So this is the coast of California. The pink and purple colors show low iron waters. So I just want to point out here, this is Point Conception. So this is really low iron water located pretty close to the coast. And what has been shown is that diatoms will become more silicified in iron-limited waters. So if you have iron-limited waters, these diatoms may take up more silica and become more dense and may ballast the POC more quickly. So my last potential mechanism is grazing. So with grazing, you have the phytoplankton growing at the surface. You have the zooplankton that eat the phytoplankton. When they are eaten, they get formed into fecal pellets. The cells get broken up. They're fragmented. When they make it down to the bottom, they're very processed. And here are some examples of some fecal pellets. Here's a kelp fecal pellet and an amphipod fecal pellet. And I want to point out that these are cells sticking out of the fecal pellets, and also this cell is the same size as this cell, so this is a much larger fecal pellet. So also all the carbon from the cells are packaged within the fecal pellet, so the cells themselves might be empty, but all the carbon is in the fecal pellet. So to bring it all together, so these are my three potential mechanisms as to why these high pulses of carbon may be occurring. So we have the large, slow-growing diatoms get disturbed, come up to the surface, form large aggregates, and sink down. We have the ballasting of biominerals. They make the particles more dense, and they sink. And then we also have grazing of zooplankton with the fragmentation within the fecal pellets, and as they sink, they're more processed. So now I'd like to go over the structure of the rest of my talk. So I'm going to separate it into my two hypotheses. My first one, I'll go over my methods and my results. And I also have this icon to help you remember which hypotheses I'm talking about. And then I'll move on to my second hypothesis with my method and result. And for this, I have two little icons for you to follow along with. And then I'll bring it together with my discussion and finish off with some conclusion. So my first question is, are phytoplankton cells driving high POC flux events? And I thought that during the high flux events, particles that reach the abyssal depths are more enriched in phytoplankton cells and have a more diverse community composition. In order to understand this, I had to get my samples. So from Station M, I collected my samples from the sediment trap here. These sediment traps are moored down at 4,000 meters deep, and I am looking at the ones that are 600 meters above the bottom. So how did I pick my samples? Here is the total POC flux over the time series, and I highlighted the samples that I chose. I chose these high flux events, which are in red, and I also chose events that occurred right before and right after in the blue. The reason I chose the before and after is I wanted to see if there was any evidence of a mechanism that could be causing this high flux event to occur. I also decided to lump the before and after events together during some of my analyses to use it as a way to see what a normal flux event would look like in this area. So now that I have my samples, I used a lot of microscopy to analyze what was inside of them. So how did I count my samples? Well, this is a 1 square of a cedric rafter shown under a light microscope. One of these squares is 1 microliter. So you would start in the top corner, and as you went along, you would pick out any cells that you saw until you got through the whole square, and you pick out all of them. So I've highlighted some of the larger diatoms that I would see in my samples, but there are actually even more in here. So I have one down here, a couple of nanoflagellates, and not only did I have to do this on 20x, I did it on 40x to look for the little coccolithophores that were harder to see. I also, so when I counted 1 square, I'd move on to the next, and I'd count 400 cells until I reached 400 cells, and then I would plug my data into a statistic by Chow et al., and that would tell me the percent of the potential species that were seen in the sample. And once I reached 80%, I said that that was enough to say there was enough diversity in the samples. So I just want to go over some really fun micrographs. So here is a large diatom called Stephanopixis, and as you kept counting, you'd find some even crazier looking cells, Rhizosolenia, and then this one is called Fragulariopsis doliolus, and then these are some Coccolithophores, and these are really tiny, so they're much smaller than the diatoms, and to tell it's a Coccolithophore, you can find the little coccolites by the refraction of the light on the microscope. And this one is called Maliania, and then this is another Coccolithophore called Helicosphaera. And then my last one is a dinoflagellate called Dinophysis. This is one of the only dinoflagellates that I was able to see because these were more rare cells. So now that I've counted all my samples, I wanted to model the carbon within the samples. So this is a graph from Mendendorf and Lessard that shows cell volume on the x-axis and cell carbon on the y. So what this is showing is that as you increase cell volume, you increase the carbon content of the cells. So larger cells are going to have more carbon in them. So in order to measure the amount of carbon per cell, I would take a picture of a cell. So this is a centric diatom, and I would measure the width and the length, and I'd plug these measurements into a volume for a cylinder. And once I got the volume, I would plug it into the equation right here, and I would get the picograms of carbon per cell. And then once I got the picograms of carbon per cell for each cell type that I counted, I then multiplied by the cell number flux to get the flux of the carbon flux for each type of cell. So I've shown you this graph already. To remind you, it's the POC flux at station M with my samples highlighted, red as high and blue as before and after. And now I want to show you this graph, which is the cell flux, cell number flux. So how many cells were sinking in each sample, with the red being the high flux events and the blue being the before and after. And it's pretty similar to the previous graph, but it's different data. So this is showing most of the high POC flux events had more cells in them, which is important for later in my study. And then now that I looked at the cell number flux, so particles will contain more cells during the high flux events. And so now I looked at the cell numbers. I broke it down into the functional groups for each of the samples. And what I want you to see from this graph is the majority of the functional groups were nanoflagellates, the red, Coccolithophores, the yellow, and diatoms, the blue. So the majority were these 3 types, so that's what I focused most of my analyses on. So I know what the cell number flux is, and I know the main functional groups. So now I want to look at the community composition within my samples. So this is a MDS plot. I did a Bray-Curtis dissimilarity matrix, and I plotted it on the I plotted it on an MDS plot. So the axes on an MDS plot are arbitrary. It's mostly the grouping of the samples. And I want to point out that one point is the community composition of one sample. So it's not different types of cells, it's the whole community of one sample. And what I found was that when I ran a permanova on the high and the before and after, I found that there were two distinct community compositions, the high flux and the before and after. I then wanted to look at the diversity within my samples. So I ran a Shannon diversity index, and what I found was that there was no difference in diversity between the high flux and the before and after. I also want to point out that the average annual Shannon diversity is around 1. So my samples are actually more diverse than what you would find in surface waters in this area. So now I have the community compositions, and I wanted to see what was driving the differences in the communities. So these are 3 graphs that show the relative cell flux of 3 main functional groups. So my diatoms, my coccolithophores, and my nanoflagellates. My y-axis is showing the number flux normalized to POC. So why did I normalize my data? Like I had shown you in that second graph, most of the high flux cells had more, high flux events had more cells in them. So I wanted to look at more so of the enrichment of the particles rather than just the numbers. So what I found was that in each of the 3 functional groups, there were less cells sinking in the high flux events. This is saying that the particles are less enriched in the phytoplankton cells in the high flux events. So now that I know that there are less cells sinking, I wanted to look at the carbon within each sample to see if it matched up with the cells. So this is my total carbon normalized to POC with my diatoms, coccolithophores, and nanoflagellates. And what you see for the coccolithophores and the nanoflagellates, they both had less carbon being transported down during the high flux events. But for the diatoms, we say that they're bringing down the same amount of carbon in both high flux and in the before and after events. So this is showing that diatoms there must be something in the high flux events that's causing more carbon to be brought down. I also want to point out that this is my modeled carbon per cell, and my modeled carbon was the theoretical maximum of what could potentially be in the samples. And so these are fractions. So we can see that this is much higher than it should be, and I just want you to be aware of that. I will talk about that in my discussion as to why this might have occurred. So now that I see that this high flux, there's something going on with the carbon being brought down, I looked at the individual cell types to see what was driving this. So these are 9 different types of phytoplankton that I had seen in my samples, and these were all significantly less enriched in the high flux events. I also want to point out that these are pretty small, different types of cells. I did find, however, that this one large diatom called Rhizosolenia was higher in the high flux events. So I plotted the mean carbon flux of the high events divided by the before and after events to show how much more carbon Rhizosolenia was bringing down than the other ones. So this red line here is showing 1. So everything below 1 is showing that these were all significantly lower in the high flux event. But what I want you to take away from this Rhizosolenia is it's bringing down almost double in the high flux events than in the before and after. So they're bringing down a large amount of carbon. And I also want to address this very large error bar. So here is the data for Rhizosolenia. You can see that most of the data in the high flux events are approximately 10% of the total POC sinking down. So there are these outliers that are 60, 80, 100%. So those are what's going to be affecting my error bars in that previous graph. So now that I know Rhizosolenia has an impact on the high flux event, I decided to group my data based on size. So this again is my carbon normalized to total POC flux for my large cells, which I categorized as having a volume greater than 1,000 micrometers cubed. and my small cells, which I categorize as having a volume less than 1,000 micrometers cubed. And what we see is that the small cells have relatively less carbon in the high flux event, but the large cells are still bringing down the same amount of carbon in the high and in the low. So bringing it back to my question, are phytoplankton cells driving high POC flux events? So what I thought was that during the high flux events, particles that reach abyssal depths are more enriched in phytoplankton cells and have a more diverse community composition. Actually, that's not what I found. I found that high flux particles are less enriched in phytoplankton, and they do have a different community composition. What is also interesting is that I found that these large diatoms are driving some sort of change in the high flux events. So now I'd like to move on to my second question. Are high POC flux driven by ballasting of cells? And my hypothesis is that biogenic silica and calcium carbonate in sinking particles are positively correlated with POC flux during the high flux events. So in order to address this, I had to measure the biogenic silica in my samples. So how do you measure biogenic silica? First, I filtered my samples on a 0.2 micron polycarbonate filter in triplicates. Those filters were then put into an oven for 24 hours to dry. Those filters were then put into a Falcon tube for the analysis. So the analysis is taking a strong base, NaOH, and submerging the filters in them. They were then put into a hot water bath for an hour to ensure that all the silica was dissolved within the samples. I then added ammonium molybdate, which will turn the samples them different colors, different shades of blue, as you can see here. And this absorbance was measured on a spectrophotometer. And I just want to point out that the darker the blue, the more silica the samples have. So I measured my biogenic silica and I plotted it against POC. So this is BSI flux and POC flux. Now, I also am showing my PIC flux. The PIC, so particulate inorganic carbon, was given to me from the Smith lab at MBARI. And what you can see here, and so BSI, again, comes from diatoms, and PIC is Coccolithophores. And what you can see here is that as you increase POC, you have an increase in BSI, and the same thing for PIC. So they both have a positive linear correlation with POC. But what I want to draw your attention to are these 3 dots over here on the PIC. So it does have a positive linear correlation, but these 3 dots are corresponding to the highest POC flux measured. And you can see that they actually seem to be decreasing. So what I did was I wanted to look at the enrichment of these biominerals in the total POC. So I normalized my data, again, to the total POC, and this is what I got. So this is my BSI flux normalized to total POC. And you can see that as you have increasing POC, you have this constant amount of BSI sinking. So as you increase POC, you have increase of BSI proportionally. And then for the PIC data, what I want you to see is that there's a negative relationship happening. So when you increase POC, you actually get a decrease in PIC. So another important thing in understanding the ballasting of the cells is understanding if the cells are still alive or contain a chlorophyll when they reach the bottom. So here I have my data broken down into cells that had no chlorophyll and cells that had a chlorophyll intact when they reached the bottom. So for the cells that didn't have a chlorophyll, they brought down the same amount of carbon in high and in the before and after. But for the cells that did contain a chlorophyll, there were less cells bringing down carbon that contained a chlorophyll in the high flux events. So to bring it, so these are examples of cells that do not have a chlorophyll and cells that do have a chlorophyll. So these little light spots are what the chlorophyll would look like under the microscope. So to bring it back to my question, are high POC flux events driven by ballasting of cells? So what I thought was going to happen is that biogenic silica and PIC in sinking particles are positively correlated with POC flux during the high flux event. Well, what I did see is that BSI and PIC both had a positive linear correlation, but the BSI appeared to be having a better effect on the ballasting. And also, I found that there are fewer cells that contained a chlorophyll in the high flux events, which could mean something else is happening. So to bring it back to my first question is, are phytoplankton cells driving high POC flux events? What we found is that high flux particles are less enriched in phytoplankton, and they contain a different community composition. And also, really important, that these large diatoms are driving this change in the high flux events. And my second question is, are high POC flux events driven by ballasting of cells? And what I found here was that the BSI appears to be driving the ballasting in the high flux events, and also that there are fewer cells containing a chlorophyll in the high flux events. So now I'd like to bring it all together in my discussion. So I want to go back to my potential mechanisms that I had addressed in the introduction. So my first potential mechanism is the aggregation of large diatoms in physics. So like I had stated before, these are the large, slow-growing diatoms that when they get disrupted, they get brought up to the surface, they get stressed out and form aggregates. These aggregates will sink down to the bottom. So which of my results would support this? Well, here, as we look back at the satellite images, we know that upwelling is occurring here. So if upwelling is occurring here, there might be those large diatoms that are getting brought up back to the surface. Those large diatoms are being brought up to the surface, and they're moving along this front. And this front is a known high export of carbon. Those large diatoms might be making it down to the deep ocean through those aggregates in this area. And these, I want to point out, are the rhizosolenia cells, and they are much larger than most of the other cells that I saw in my samples. Now, going back to my second potential mechanism, the ballasting and biominerals. So here I have my biomineral-rich phytoplankton that are getting eaten. They make their fecal pellets more dense, and they sink down. But I also want to go back to the iron limitation that I had talked about in the introduction. Like I had shown, that California, the water really close to the coast of California has low iron. And if diatoms are growing close to the coast of California, like I had shown in the previous slide, they might be iron limited and taking up more silica. They're taking up more silica, they'll become more dense with this biogenic silica and make the particles even more dense when they sink. So what supports this? This is, again, my biogenic silica to POC normalized. And as you see, you have as you increase POC, you have an increase in BSI. So we did see that BSI and PIC were both positively correlated, but BSI is showing this better correlation to POC, which is contrary to what is globally seen. What they normally see is that PIC has a better relationship to POC than BSI. But if the California current is iron limited, it could make sense that these diatoms are containing more biogenic silica. And then moving on to my third potential mechanism, which is grazing. So again, we have the phytoplankton growing at the surface. We have the zooplankton feeding on them. As they get broken down into the fecal pellets, they get fragmented, and they're very processed when they reach the bottom. So what supports the grazing hypothesis? The chlorophyll content. Well, we saw that there were less cells that had a chlorophyll in the high flux event. If that's the case, they must have been more processed, or there must be something that the high flux event cells are going through to lose their chlorophyll. And also, a lot of the cells were highly fragmented, as you can see in this picture. If they're highly fragmented, there must have been a mechanism like grazing that could have caused them to fragment and break up. And I also want to address the overestimation of the modeled carbon that I had talked about. So these are the large diatoms, rhizosolenia, and you can see that they're very fragmented. And as I had shown, these rhizoselenia bring a huge amount of carbon down during the high flux events. So if these are fragmented, these are large cells, they might have been double counted. They also might not be containing carbon because their cells are fragmented. And those could be reasons why the model carbon was much higher than expected. So now I'd like to bring it back to all my potential mechanisms. So again, we have these slow-growing diatoms being brought up to the surface, forming large aggregates and sinking down. We have the biominerals and the ballasting of the cells with low iron, which could mean more uptake of silica, denser particles that sink down. And then we also have the grazing of the zooplankton with the fragmented of cells where they lose their chloroplasts, and when they reach the bottom, they're more highly processed. And we don't know the exact mechanism, but I think it's a mixture of all three. So some future work that could help resolve this could be the genetic data alongside the microscopy. So like I had said, there were a lot of fragmented cells. So the genetic data could help tell me what those fragments are. The thing with genetic data, it won't tell me how much of what's sinking, and it's not always the most accurate. Some genetic work has shown that there haven't picked up a lot of Coccolithophore DNA samples, and I found a lot of Coccolithophores in my samples. So the genetic data will help fill in the gaps, but we still need microscopy to help get an accurate picture. Also understanding the differences between aggregates and fecal pellets. If we can understand if it's an aggregate that's sinking or a fecal pellet, we can try and see if we can resolve the mechanism as to why these high flux pulses are occurring. And also looking at satellite remote sensing of the chlorophyll sinking, so being able to see where the chlorophyll at the surface and how it sinks down to the deep ocean. And with that, I would like to say thank you to my committee members. Thank you, Colleen, for helping me with everything. You've been fantastic. Tom, for just always being there whenever I have questions about statistics. And Ivano for being on my committee and just being a great asset to me. And Ken for basically giving me my project. My samples came from you, so thank you for that. I'd also like to say thank you to Chrissy, who helped me go through the freezer and collect all my samples. My friends, Sean, Felicia, Mia, and Emily, for just always being supportive and going through all my practice talks with me. My parents for being supportive across the country, and Ben for always just being there for me. And then there's a whole bunch of you that I didn't mention by name, but the whole ML community have been so helpful here. And I just like to show some great pictures of my friends here, so yeah.
With the graphs that you had that were showing biosilica to POC, you had POC on both axes. And so what's the difference between those POCs?
So for the first one, I only had the POC on the X. On the second ones, I normalized my BSI data to the total POC to get the relative amount of BSI as you increase POC.
But those were different POC numbers on axes? You didn't just divide that number by...
Oh, I did. So I took the BSI amount and I divided by the total POC for each sample. So it is the same POC on each. Is that answering your question? Okay. Okay. Yes.
Cindy, did I get it right that you said that when you took the allometric conversion of the cell measurement into its corresponding carbon content, that when you added that up from the cell counts and their size, did you say that that carbon flux exceeded the measured carbon flux?
Yes. Yeah. So the amount that I calculated, modeled, was much higher than was actually sinking for the total carbon.
Has that ever happened?
I don't know. But I checked my calculations, so I think it must be something to do with those really large fragmented diatoms that could be affecting it. Yeah.
And wouldn't that suggest more of an impact on grazing, where it would take away that chlorophyll?
Yeah, so we don't really know the mechanism, but it does support grazing if you have a lot of this fragmentation and a lot of the chlorophyll that isn't seen in those high flux events. So there must be more grazing occurring during high flux events. Yes.
Would you say that in each of the samples you counted, you were able to identify the majority of the cells, or were a lot of them fragments and like things that were broken up and digested?
So yes and no. So like there were a lot of samples that were just a whole bunch of fragments that were just really hard to depict what they were. But I would say most of them I was able to at least see what the fragments were. Like there were some defining features of them. Yeah. Yes.
So silica,
Yes.
in seawater is highly unstable, gets dull as a and it's very selective in size and shape. How do you think that plays in the calculation?
So
Like, talk about upwelling tests, diatom tests, they get dissolved as they precipitate to the water column.
yes.
The larger, less, and the smaller ones, the larger, how they
So like how would they lose the biogenic silica as they're sinking? Am I understanding the question right?
selective dissolution of
That's a good question. I guess, so how does it, let me think about that for a second, sorry. So like, I guess if they're losing their biogenic silica as they're sinking, it could mean that they're not making it as quickly down to the deep sea. So I guess the ballasting might not be as effective as they're sinking. I'm not 100% sure, but I can look this one up and see what happens. Yeah. Yes.
Do you think the collection method caused some of the fragmentation? There's something about
There could be. So these were, yeah, these are also older samples, so a lot of them were pretty old, but and they were frozen and thawed and refrozen, so that could also affect the fragmentation of the samples, of the cells, yeah. Yes.
Okay, I've been thinking about the graph.
Uh-huh.
It took a while. And it doesn't look like biosilica was increasing with increasing POC. That looked level.
Yes, so
there's no change.
So if there's no change, it would mean that as you're increasing the total POC, the amount of BSI is going up proportionally. So as you're increasing so if it's a fraction of the amount of BSI to the total POC, you have this increase in total POC, and if it increases proportionally, it's not going to change the ratio. So it's a ratio between BSI and POC.
So it's unchanging, it's not that it's increasing.
Yeah,
like
yeah,
it's
that's,
just
it's
there's more
more,
carbon and more silica.
Yeah.
in even amounts.
Yes.
Okay,
yeah,
got it. That's good.
okay. Yes.
You showed that global data set by Claus and Archer, which was just PIC versus POC without doing that normalization. Has anybody else looked at that in that way? Did they
Normalize
it in order to?
I don't think I saw anything about normalizing biominerals, but the reason I normalized it was to just see how like increasing POC, just looking at the relative amounts instead of looking at the total amounts, because when you have more of the POC sinking, like I had shown with the cell flux, you had more cell fluxes, so you might have more biominerals. So I kind of wanted to not just show the absolute numbers, but the relative numbers.
Increasing because everything's
Yeah.
or is
Is it increasing?
PIC or PSI becoming
Yeah. Mm-hmm. Yeah. Yes.
So one more on that, the thing about that, counting the cells exceeded what was measured.
Mhm.
So, did you consider measuring the actual chlorophyll in the trap?
Like the total,
The
chlorophyll.
extract, well,
Oh, like looking at like the extraction of chlorophyll in the samples?
yes.
I did not look at the chlorophyll, but I did take note when I did see a chlorophyll in a cell when counting the samples, so.
I had a feeling your answer to that one was to be no. Because
Yeah.
You just didn't do that.
Yeah.
So, of the ones you counted, of the things that look like cells,
Mhm.
what was more abundant? Hollow cells or cells with gook on the inside?
Hollow cells and a lot of fragments, so like they didn't contain a lot of chlorophyll. Yes.
How did you deal with fecal pellets? You like tear them apart to find out what?
I didn't look at the fecal pellets. So I looked at what was basically like could have been inside a fecal pellet. So my samples were just like the liquid part. Like, yes.
I want to say this is the most questions I've ever heard in a thesis defense here. But you did a great job.
Thank you. Yes.
Going back to somebody else's question, talking about like the freezing and thaw.
Yeah.
Because I know some of your samples were from a really long time ago. How
Yeah.
long was the time series, and do you think that could have had an effect? Like the older samples maybe were more fragmented than the newer samples.
Like, did I see them more fragmented as a?
Yeah, or even just like how long ago were some of these samples taken? This is a pretty impressive data set that you've got to use. Right?
Yeah, the earliest is 1989, but I didn't look at those. I think my earliest is probably early 2000s that I was able to look at. Yeah. Yes.
When you did your community composition for NMS
Yeah.
plots, did you see any relationship with time? Like, did you
Oh,
look for finding those points?
I didn't do it against time. That'd be something interesting to see, though. Yeah, I haven't. No, not from this data. Yeah, that would be interesting to see. Yeah, that would be cool. Yes.
Why do you think there's been this increase in the last decade in those high episodic pulses?
Could be from like increased carbon in the atmosphere. So if you have more phytoplankton uptaking the carbon, they could be transporting more of that down. But yeah, that's my guess for that.
So increasing news of the phytoplankton?
So I guess it could be increased blooms, but like just having more carbon in the atmosphere would be absorbed, like be dissolved in the ocean, so it would make more carbon available to the phytoplankton, so that they could sink with more carbon. Yeah.
Any other questions? Another chance.